enzymes often come in sections. So the main part of the enzyme is what's called the apoenzyme, and it's made up of a protein, but it can't function as an enzyme because it's not complete. It needs to have cofactors. Now, cofactors can be either inorganic. This is probably an atom of iron or magnesium or mobilium or something like that. And if you have something that is organic but not a protein that is important to the functioning of the enzyme, we call this a coenzyme. A coenzyme is an organic cofactor, whereas oftentimes if we say just cofactor, we're referring to the inorganic cofactor. Now in this particular enzyme, once we put the cofactor in and the coenzyme into the apoenzyme, we get the hollow or whole enzyme, and we have an active enzyme. Now this spot here is called the catalytic site or the active site. So on this diagram here, we have the enzyme, and this one has two active sites. The substrate fits into it perfectly, and the active site does whatever the enzyme is going to do to the substrate. So you can think of the rest of the enzyme as just getting the shape of the active site. Now let's talk about enzyme activity. So here we have a space-filling model of an enzyme and a substrate. It's a little more detailed, a little more accurate than our cartoon that we had on the last slide. So here's the active site, the substrate comes in, and look at that! We changed shape. This active site is not the same shape that it was over here. We call this change of shape a conformational change. What happens is the binding of the substrate to the active site causes this conformational change, and this conformational change enables whatever the enzyme is going to do to the substrate to happen. So we either have two substrates that are being mushed together. You can kind of see how that would act like kind of a pincer to smush them together, or you can see how it might be a scissor action where the conformational change causes a breaking of the substrate of the molecule into two separate substrates. Here's a graph showing the action of an enzyme as it relates to activation energy. So let's talk about what activation energy is. Whenever you have two reactants that are going to break down into a product, okay, this is an exergonic reaction, energy is going to be released. Here we have energy here, progress of the reaction. So the reactants have more energy than the products. So this is a spontaneous reaction. But it takes energy to break the bonds that are holding the reactants together. We call this the activation energy. Well, the activation energy, as you can see here, is much higher without an enzyme, whereas with an enzyme, you have a much lower activation. The current theory, at least the current favorite hypothesis, is that this conformational change stretches the bonds, pulls the atoms apart, moves them closer together, and that lowers the activation energy. That's why reactions proceed more consistently and faster with an enzyme than without. When we're talking about the activity of an enzyme, there are several things that control the activity. And one of these is temperature. All enzymes work best at a certain temperature. Remember, temperature is a measure of the movement of the molecules or the atoms. So if we increase the movement, increase the temperature, if we put energy into the system, cause the enzyme to vibrate more, it may work better and faster. We'll come back to that in a minute. pH is another one. pH can affect the active site. In many enzymes, in the active site, we have amino acids or an amino acid that has an ionic side chain. So that means it's either acidic or basic. So let's go with the example of, say this enzyme has a key amino acid in the active site that has a basic side chain. And this substrate happens to be acidic. And they are attracted to each other, and that's what causes the reaction to take place. So if you change the pH, maybe this base becomes neutral, and then the enzyme doesn't function anymore. So all of these together influence the functioning of an enzyme. Now, if you get too high of a temperature, so we'll say we've got a graph, and we're going up in temperature, and the activity of the enzyme is going up and up and up, and all of a sudden it crashes to 0. Well, what's happened? The temperature got to the point where the vibrating of the various atoms caused the functional protein to unravel, and we get what's called a denatured protein. So this is what happens when you cook an egg. A raw egg, the white is clear and runny, the yolk is runny, and it's more opaque yellow. But once you cook it, it becomes a solid. And the whites go from clear to white, that's why we call it the white. The yolk becomes less clear, it becomes very solid, and it's because we're taking the proteins in the egg and we're denaturing them through heat. We're unraveling them, and as they cool off, they're no longer functional because they don't go back to the original form. That's part of why we cook things. Because, getting back to microbiology, instead of just cell biology, when you cook your foods, you denature proteins within the bacteria and you kill them. That's why you can raw meat will rot faster. You have all of these environmental bacteria, nice moist meat, and they're growing happily. But if we take that raw meat and we cook it good, then we kill all of those bacteria. We also dehydrate it. We'll talk more about that under control of microbes. But that's part of why we cook things, and that's how we control microbes. And it's because of the effect of temperature on enzymes. It's also structural proteins. But the enzymes work faster and faster and faster. They reach their optimum temperature, and then they denature, and you don't have that enzyme anymore. And either you've got to make a new one or you die. Now, pH will do the same thing, but it's a little different. Instead of vibrating this until it comes apart, say we've got ionic bonding going on in here, holding these beta pleated sheets together. If we say lower the pH so that we've got all sorts of extra hydrogens around, we're going to lose the ionic bonding here, and it's going to unravel. Same thing goes for here. Suppose we put a base in and we decrease the number of hydrogens. Same thing's going to happen. You're going to mess with your ionic interactions and you're going to denature your protein. You can also cook with acids. Ceviche, I probably said that wrong. Basically, it's a dish where you take raw fish and you squeeze lime or lemon juice on it. You're denaturing the proteins, you're reducing the microbial content. We don't cook with bases because you can't eat them afterwards. If you eat a base, it denatures your esophageal lining and we don't do as well. These regulate activity. Now, before we move on, different enzymes have different temperature and pH requirements. The enzymes in your body are set up to work at around 37 degrees Celsius, body temperature. Some of your enzymes work better when you have a fever. These are enzymes that are involved in fighting the infection. Makes sense. But there are bacteria that we've isolated from carcasses of whales off the bottom of the ocean. There the temperature is 4 degrees C, just above freezing. We take the enzymes from those microbes, and that's their optimum temperatures. We get them much above 4 degrees C, and those proteins will denature. You're going, why is she talking about whale carcasses? Well, because we take these enzymes from these cold water microbes, and we take the ones that break down fats and break down proteins, and we use them in cold water detergents. Isn't that cool? Microbes supply us with everything that we need. Now, pH. You have an enzyme in your stomach, pepsin, that breaks down proteins, and it works best at a pH of between 2 and 3. Now, when the contents of your stomach leave your stomach and go into your intestines, your liver, through the process of your gallbladder, put in biosalts and change the pH of the food bolus from acidic to just under neutral. Well, pepsin stops working. It doesn't work at a near neutral pH. Your body produces another proteinase that works at near neutral pH called trypsin. Now, would trypsin work in your stomach? No, it would denature. Pepsin doesn't work in your intestines, not necessarily because it's denatured, but because it needs that pH for the active site to work. So hopefully that helps you understand this portion. Now, there are ways to control enzyme activity other than through temperature or pH. One of them is through competitive inhibition. If your body wants to slow down the action of an enzyme, suppose we've only got a few of the substrates and we don't want to use them all up all at once, then the body can make a competitive inhibitor. See how that's about the same shape? It gets in there, blocks the active site, the enzyme says, hey, wait a minute, spits it out, now the substrate can come in. Basically, because the inhibitor and the substrate are competing for the active site, then it slows things down. Now, we can reverse this by increasing the substrate concentration. Suppose the cell just imported a whole bunch of the substrate. Now there's more substrate, it's more likely to get into the active site when you have the same amount of competitive inhibitor. Now this is called reversible competitive inhibition. The inhibitor comes out of the active site. There are some poisons that once they get in the active site, they don't come out. We call this non-reversible competitive inhibition, and it kills the enzyme. Keeps it from ever functioning again. So the cell has to break down that enzyme and make a new one. Well, if you get enough of that poison, then you die. Now, some wild mushroom poisons are in this category. They are non-reversible competitive inhibitors. So be careful when you're stalking the wily mushroom out in the wild. Now, another way to inhibit or activate an enzyme is through another site called the allosteric site. It's not the active site. The allosteric site is strictly for regulation. So here's an enzyme, it's got an active site, it's doing its thing, but say we've run out of substrate, or we don't have enough, or we don't need the product. So we take an inhibitor, put it into the allosteric site, and it causes a conformational change. Changes the shape of the active site so the substrate doesn't fit in. Basically, we've turned off this enzyme without resorting to competition in the active site. Now, we can also turn on an enzyme. Suppose we've got an enzyme that we only need to put into action occasionally. In its normal state, the active site does not fit the substrate. But when it's time, we can put in an allosteric activator, conformational change again, the substrate fits into the active site, and we've basically turned on the enzyme. Now, here's another way, or another example of allosteric inhibition that we call feedback inhibition. So, most of what goes on in the cell involves enzymatic pathways. We start out with enzyme number 1, takes the first substrate, does its thing, creates intermediate A. Enzyme 2 takes intermediate A, makes intermediate B. Enzyme 3 takes intermediate B, makes the end product, which goes on to do whatever it's going to do in the cell. Once we get a high enough concentration of the end product, oftentimes it'll go back to enzyme 1 and it'll bind to the allosteric site and turn the whole thing off. Here's a non-cellular example. Suppose you are the owner of a bicycle factory, and your employees are really good at their job and they're making bicycles, and pretty soon your warehouse is full. Pretty soon you're stacking up the bicycles in your office, and then you're stacking them up in the front waiting room, and then you're stacking them up into the employees' break room. And you say, okay, no more bicycles, we gotta stop. So we shut the assembly line down. And it's better to shut down the assembly line at the beginning as opposed to having parts of bicycles sitting around. You either want all of the parts in their little bins, or you want them to be a complete bicycle. Then you tell your employees to go home and promote your bicycles. Tell everybody that they got to buy bicycles for their kids. Well, once you sell off the bicycles, then you start your assembly line up again. Once the concentration of the end product drops, This drops off, the enzyme turns on, and we start making that product again. Hopefully, the assembly line analogy worked for you. Now, here is an actual example of some metabolic pathways that are involved in making different amino acids. We have tyrosine, phenylalanine, and tryptophan. So you take erythrose 4-phosphate and phosphoenol pyruvic acid, or PEP, and you have different enzymes that can take these, mess things around, and make these 3 amino acids. Well, you can see that phenylalanine can go up and turn off synthase 2. It's involved in making phenylalanine, so you would only want to turn that one off when the concentration of phenylalanine increases. If you get a concentration of tyrosine, you don't want to turn off synthase 2. You want to turn off synthase 1. Same thing with tryptophan. See how that works? Okay, that's it for this topic. Whew, that was a long one. This is kind of a dense chapter, so that's just to be expected. So here are reminders of what I want you to learn from this. And next we will talk about carbohydrate catabolism, in other words, glycolysis, and aerobic respiration.